sir.
Okay.
Rahman, sir.
Yes. Thank you.
Thank you, sir. Next, I
So,
extend my warm madam for her wonderful support to arrange this 102 International Virtual Learning Series. Thank you, Dr. Irfan, madam. Thank you, madam. Here for today's session. So, session will be a good platform for the students to get a good knowledge. This is the first and foremost needy for your career. So kindly utilize this great opportunity to explore your knowledge. So all the very best, my dear students. Once again, I welcome you all. Thank you. Professor Dr. Abdur, sir. Thank you, sir. Most welcome you, sir.
okay, you're welcome too.
Dr. Shivakumar. Dr. Shivakumar. Hello. ஐயோ அப்போ கொஞ்சம் இது ஓகே ஓகே தேங்க்யூ தேங்க்யூ தேங்க்யூ
Thank you. Good afternoon. Our 102, 102 International Virtual Learning Programs. Honorable Convener, Professor Dr. N. Venkatesan, Principal AKCP, Organizing Secretary Mr. Arthan Ari Shawen, Associate Professor AKCP, and participants, ladies and gentlemen, good afternoon. First of all, I would like to thank the AKCP authority to provide such a wonderful opportunity and honor to be a speaker. Before starting the presentation, let's introduce myself. Already it is introduced. Already you know about these things. I'm Dr. Aziz Rahman, Professor of Department of Pharmacy, University of Rajshahi. Actually, University of Rajshahi is situated near the border of West Bengal, Paschim Bang, Bangladesh. As you know, I did PhD from Kongju National University on 2007, postdoc from NCNPO, Mississippi, and postdoc from KRIB, Korean Research Institute of Bioscience and Biotechnology, Daejeon. Daejeon is a science city in Korea. So my research interest is isolation, purification, and characterization of bioactive natural products, especially anti-cancer, anti-inflammatory, antibacterial, antioxidant, antidiuretic. It is a component from plants, also from mushroom, microbes, using Hplc, GCMS, NMR, and other spectroscopic techniques. Also, nowadays, I am working on an interesting topics, prebiotics and probiotics, especially lactobacillus, probiotics found in milk. Anyway, today I am planning to present a research work related to natural product. I mean, the path by which a natural product chemist achieves his or her goal using example of work done in Korea. Basically, that's my PhD work. So, the title of today's presentation is Phytopharmacological Properties of Investigated Korean Medicinal Plant. So, what is phytopharmacology or herbal medicine or a natural product? Since I work and still working on natural product, at first we need to know the theme of natural product. Basically, natural product is the term used to describe any product or any medicine or drug that are derived from nature. Nature means plant, animal, microbes, or mushrooms, or any other from nature. Herbal products or herbal medicines, also it is also called herbalism, is one of the natural products where we learn in pharmacognosy the use of herbal medicinal plants. So in herbal medicine or herbal natural product, we learn the use of herbs or herbal medicinal plants. It is also called phytomedicine. It's very popular in developed and developing countries due to effectiveness and less toxicity. So I put some pictures of herbal medicines commonly used Indian herbs. Right side, I think in Tamil Nadu, I think this is in Tamil. Anyway, this is very famous in India, and also Korean ginseng. They are all herbal products. The popularity. According to WHO report, 80% of the world population use herbal medicine for treatment of various diseases as well as for prophylactic use. And also in India, more than 70% of the Indian population use herbal drugs for their health. And Indian herbal drugs are also famous in Bangladesh too, especially Amloki, Ashwagandha, Brahmi, Nimba, as well as Tulsi. And in Korea, around 70%, 69% people use traditional medicine. In 2010, Korea exported around 0.8 billion herbal medicine. Huge market. Herbal market is a huge market. So I put two pictures. The left side is from the plant Vinca rosea. Nayantara, it's called in Hindi. It is actually well-known cytotoxic drugs available in market, and these drugs, these two, vincristine and vinblastine, these drugs are derived from these herbs, Nayantara. And India, Oiluna Pharma, I think they're selling this product. And we know vincristine and vinblastine are very famous anti-cancer drugs that are treated currently against acute leukemia, brain tumor, thyroid toxicity, antithyroidal drugs. Right side, another plant, yew, Texas derifolia, and well-known anti-cancer drugs nowadays available in market, Taxol, Practitaxel, generic name, which is used in ovarian cancer, breast cancer, lung cancer. So this plant, this compound Taxol is derived from also natural product, herb. Now, let's say something about herbal medicine in Korea. Traditionally, Korean medicine is widely practiced in Korea, and actually it is originated from traditional Chinese medicine, And later, for a certain duration of time, Japanese medicine, but basically originated from traditional Chinese medicine. And it was practiced in prehistoric time. Prehistoric time means it was traced back as far as 3000 BC, before Christ. That means a long history, Korean traditional medicine. Also, our Indian medicine has a long history too. So presently, Korean medicine has become a part of national health system, and by integrating traditional and modern approaches, both approaches, I mean Western allopathic medicine, pharmaceutical medicines, and traditional Ayurvedic or herbal medicines. Nowadays, Korean medicine constitutes many complementary and alternative approaches that are continuously developing and validating. And as I already mentioned, more than 69%, around 70% people are depending on this herbal medicine. In Korea, there are a variety of herbal markets, well-decorated herbal markets. One of the famous markets is Seoul Yongsan Market. It was established in 1960th century. A lot of plants, herbs are available in this market. And currently, Korean herbal medicine is practiced by 20,000 certified clinicians using especially acupuncture and medicinal herbs. And it takes around 10% of national health insurance. A number of universities in Korea are now focusing on Korean traditional medicine, or it is also called Oriental medicine. Huge number of foreign and local students are enrolling there. I saw a lot of Indian students too there. So we are discussing about herbal medicine. It is also called Oriental medicine. Very popular term is traditional medicine or folkloric reputed medicine. All belongs to natural product science or chemistry. So herbal medicine in called Oriental medicine department or herbal medicine department, traditional medicine department. All belongs to same theme. Can you hear me? Hello? Okay, good. Good. Okay. So now let's see introduction. The objective of this presentation. Today's objective of this presentation is, today's presentation will explain what natural product chemists want and how they reach the goal. The process includes selection of herbal plants having folkloric reputation in Korea, activity tests, our desired activity test, what we wanted that time, antioxidant, antimicrobial, and cytotoxic, isolation of desired components, phytochemicals, identification of the isolates using conventional spectroscopic techniques, and finally activity test of the isolates, desired activity test. Test, antioxidant test, antimicrobial test, and cytotoxic test. So we selected at first. Here you can see the flow chart, flow diagram of my work, my PhD work. Actually, this is general diagram for anyone who wants to work on herbal medicine or natural product. At first, we search a huge number of plants for a specific pharmacological activity. Let's say, for example, antioxidant activity. So we search plants from Korean origin, plants available in Korea. We found thousands of plants. From the thousands of plants, from using literature review or other, we sorted these plants into certain numbers. And what we did, after sorting the plants having folkloric reputation, we selected few plants from mountains. In Korea, more than 60% area are covered by mountain. And a huge number of plants, mushrooms are available in this mountain area. So we went mountain, riverside, roadside, and sometimes sometimes forest to collect specific plants. And then we did these plants roughly. We did extraction using 80% aqueous ethanol, sometimes methanol. This is 80% aqueous ethanol is popular. And then after extraction, we did in vitro screening. My goal was antibacterial, cytotoxic, and antioxidant. So we did screening. against these three activities. Okay, after getting good results, we purchased the plant from that herbal market, Seoul herbal market, as well as Daejeon Science City herbal markets on large scale. After that, we did extraction, and then we proceed with chromatographic techniques. There are lots of chromatographic techniques. We will see this later. And then to isolate pure compounds. Pure compound means to isolate compound responsible for that particular activity. Let's say antioxidant activity. The plant shows antioxidant activity. So we will do column chromatographic techniques. We will isolate compounds, pure compounds. And from these pure compounds, we will find which compound shows antioxidant activity. That compounds will be analyzed spectrophotometrically to determine its structure, spectroscopically and other spectroscopic techniques to determine its structure as well as activity. So this flowchart is called bioassay guided isolation of active constituents from natural products. People use herbal medicine traditionally, but that herbal medicine must contain certain components which are responsible for that activity. Let's say, for example, one compound shows on herb shows antioxidant activity. What does this mean? This means it has some active constituent. That constituents are responsible for that antioxidant activity, not whole components of that herb. A herb contains million compounds. From there are some particular compounds responsible for that antioxidant activity. So let's go ahead to discuss more. Selection of plants. From thousands of plants, we selected 215 plants, Korean plants, Korean origin, for 3 assays: cytotoxicity assay, antifungal assay, and antioxidant assay. Cytotoxicity assay was performed against A549 cell lines, that is called actually lungs carcinoma cell lines, SKML2 skin carcinoma, and B16F1, which is mouse melanoma cell line. And then antifungal assay, trichophentonmentophytes, it's skin and scalp infection it causes, and Candida species that causes fungal urinary tract infections and other infectious disease. So cytotoxicity assay, antifungal assay, and then we performed antioxidant assay. Roughly, antioxidant assay was done by DPPH free radical scavenging assay and ferric reducing antioxidant power assay. From 215 plants, we found 10 plants that showed satisfactory results. Right side, just I put some pictures of plants that I used in this experiment. So from 10 plants that showed satisfactory results, we choose 3 plants. Why? From literature review. The plants whose literature review was less, or we didn't find any good literature review, was chosen. And actually, the natural product research continues to really challenging job. Why? Because it depends on your faith as well as on your working way. Sometimes you will be successful and sometimes you may not. So here we selected 3 plants: Dendanthema jawadisci, Draba nemorosa, Trichosanthemum purulenti. Why? To achieve our goal. What goal? To get cytotoxic compounds from this plant, antifungal compounds from this plant, and antioxidant from this plant. 3 plants were actually categorized based on their activities. For example, Dendanthema jawadisci was chosen for antifungal and cytotoxic activity based on its activity data. Draba nemorosa was chosen for antioxidant and cytotoxic activity, and Trichosanthemum was particularly chosen for cytotoxicity. This is our assay result: antifungal assay, antioxidant of 10 compounds. From these 10 compounds, we choose 3 indicated by red highlight: Trichosanthemum jawadisci, Draba nemorosa, and Trichosanthemum purulenti. This is Dendanthema jawadisci. I don't know if it's available in India or not. It is an ornamental plant. available in western part in Europe, and as well as in Korea and China. It is also named. It is also called chrysanthemum, Asteraceae family. So this is traditional use. It is used in pneumonia, bronchitis, cough, pharyngitis. Anyway, these are the traditional use. In literature, we found some sesquiterpenes, flavonoids, and polyacetylene. These are this is structure are previously isolated from some else. And we found a few phytochemical works have been done on this plant, although it is an ornamental plant in Korea. So this plant Korean name is Gujolcho. Scientific name is Chrysanthemum jawordeski or Dendronthoma jawordeski. Its family is Asteraceae. We used actually root part because we didn't find any literature from root part. We found from leaf and stem part. And we collected from this from a very big Korean mountain called Gyeongsangang Mountain, which is situated in Gyeongju province. Collected in 1998. I started this work from 2003, but it was previously collected in our lab. It was in 1998, collected in 1998. These are the pictures, common pictures of this plant. Okay, what method we followed to get our desired compounds, cytotoxic and antifungal compounds? We used flash column, just freshly column chromatographic technique. A specific column, this is called open column. So first we use flash column, and then we use open column. After open column, we got some fractions. That fractions were subjected to C18 Hplc. C18, we know C18 is a reverse phase. That means polar compound comes first, then less polar, and that way carry on. So C18 Hplc. After that, I used RP Hplc, reverse phase Hplc. It was Waters Hplc system. Fractions were collected in autofraction collector. This is autofraction. Actually, all of these, I used all of these instruments in our lab. Melting point apparatus, UV spectrophotometer to determine the chromophore of isolated compounds, IR spectrophotometer, polarimeter for optical rotation, NMR. Actually, we had at that time we had an NMR from Oxford, that is 400 megahertz for proton and 125 megahertz for carbon. Mass spectrophotometer, Waters, and this is micropipette reader. Okay, this is a messy part. Actually, isolation scheme from this compound and from this plant. I isolated around 15 compounds. I indicated here 13 compounds using silica open column, as well as HPLC. This is the HPLC diagram of the isolated compounds. 9, 4, 12. This value, numeric value indicates the compound number, 12 number compounds, phone number compounds. Is just roughly. And upper side is UV spectrum of that compound. Actually, we use a photodiode array detector, PDA detector, where we easily get the UV spectrum. Okay, determination of structure. So after separation, isolation, and separation of compounds from herb, generally, we use UV, IR, mass, NMR, and polarimetric techniques to determine the structure. This is not easy process, but it's a long way to determine the structure, and also it needs a lot of experience. These are the theoretical assumptions. We need to do UV, IR, mass, NMR. These are theoretical. Practically, only using NMR, we can determine anything in this world, any chemical compound structure in this world, if we know the process. As I already told, we need this is a very advanced technology, and we need experienced person to determine the structure using NMR. but we can determine anything using NMR if we know. So this is NMR data of a compound. Just I'm showing this. It's compound 7 NMR data of compound 7 proton NMR data. This is carbon NMR data. This is carbon proton correlation in HSQC, heteronuclear single quantum coherence. Okay, this is COSY NMR data. Final structure. It's mass data. We don't need actually. This is UV data, IR data. I did everything. Okay, so from this data, from NMR, IR, UV, mass, basically from NMR, we determine the structure of these compounds. From these compounds, we found 9 compounds are polyacetylenic compounds. Polyacetylenic compounds. It has 2 acetylenic groups in its molecular structure. And from these 9 compounds, I isolated 2 new ones indicated by red border, 7E and 13Z. And there was an interesting things in there for these compounds. See, compound 7 and 13. If you look, these 2 are isomers. Same compound, same things. What is the difference? Difference is position 1 here, inside chain position 1. This is in downside and that one is upside. That means this is geometrical isomer. What happens? These compounds are really unstable compounds. So one day, I just isolated using Hplc, keep this compound, 13J, I'm talking about 13J. So I just kept that compound in near Hplc unit and left. Inside temperature is around 25. Next day, I took NMR. I got NMR data. After 7 or after 1 week, I again run NMR data of this compound, 13J. What happens? I did not find the similarity of after 7 days data and before 7 days. I mean, instantly to in NMR data. I saw some differences. Then we thought, what is the problem? First time NMR data was one type, and after 7 days, the NMR data was different than previous one. So we concluded this compound is very unstable compound. Then what happens? Actually, if this position on a number 1 position, if it gets external temperature, it changes to 7E, or 7E converted to 13J. So both are isomers but different compounds. And interestingly, one compound shows very strong antifungal effect, another compound shows less effect than the other one. So activity also depends on its structural geometry. Anyway, so we isolated 9 polyacetylenic compounds. Among these 2 compounds were new compounds. In addition to that, we also isolated other compounds, cinnamic acid derivatives. The cinnamic acid derivative is very good example and for antioxidant activity. So cinnamic acid derivatives and 1 dioxybenzene derivative. This one is a new one. So number 12 compound is a new compound, 2. So I got 3 new compounds from this plant. Okay, and then I proceed with antibacterial, antimicrobial assay. Microbial assay, first I use this diffusion assay, you know this better. And then this diffusion assay to determine the quality, and then MIC to determine the quantity. So this is just serial dilution technique for quantitative assay. And I also perform cytotoxicity assay, SRV assay. This is the flow chart of SRV assay. If you want to know, then I will discuss these things. Otherwise, just I'm proceeding to next slide. We use sulforodamine B, coloring agent, to determine the cytotoxicity activity. To determine the activity of this of isolates, cytotoxic activity of the isolates. Okay. This is the result of antimicrobial assay. We found this is anti-fungal assay. Anti-fungal assay. First one is Candida species. Second one is Trichofantum. We found all polyacetylenic compounds show good activity against fungi, almost all fungi. Number 1, 2 standard of ketoconazole, amphotericin B was the standard. So when you compare these things, this data with amphotericin B, we found number 1 and 2 compounds showed more activity than standard. More activity than standard. However, other compounds didn't show remarkable activity. Okay, this is the antibacterial and antifungal result. Upper red side compounds, this the first one shows antibacterial activity. Number 12 shows these new compound. These 3 are new compounds. 7 shows some antibacterial activity, but polyacetylene showed very good antifungal activity. This is antifungal activity against Trichophyton mentagophytes and Candida species. So cytotoxicity result and antifungal assay result is indicated in this table. Cytotoxicity assay was performed against 3 cell lines. Antifungal assay was performed against 3 Candida species as well as 1 Trichophyton mentagophytes. Actually, polyacetylene have been known to possess anti-inflammatory, antimicrobial, insecticidal, antiviral activity. And in the world, more than 14,000 different polyacetylene have been isolated from this plant. See, more than 14,000 different polyacetylene have been isolated from this plant. I also isolated a new one, two new polyacetylene. And a lot of polyacetylenes are found in Echinacea. I think it is also available in India too. And it is very famous in USA. Why? Because it contains a lot of polyacetylenes. And these are the products. And I published 2 papers from this plant. One is antifungal assay result, and another one is antimicrobial and cytotoxical. These are 2 papers. Okay, so these are all from first plant, Chrysanthemum jawadisci, Dendronthema jawadisci. Second plant, same process I applied for Java nemorosa. Plant name Java nemorosa, Korean name Kutta Z. There's some here. There's some traditional use, literature review. Yeah, isolation scheme. Similar process from this plant. I isolated around 15 compounds. Around 15 compounds. Hplc data. These are the isolated compounds. Around 15 compounds. Among 15 compounds, 3 compounds I found. New one. And cardinolides, this is synaptic acid derivatives with sugar, without sugar, and then cardinolides, sesquiterpenes, which was a new one, flavonoids. Here, there is a picture of horse. Why? Because there was an interesting story about these compounds in our lab. What was that interesting story? So this, cardinolides. The road to discover natural or phytomedicine actually is not smooth. You know, anti-malarial drug chloroquine, it was derived from cinchona bark, chloroquine. It was found as the effective anti-malarial product after investigating 14,000 similar compounds. What does it mean? After 14,000 synthetic, semi-synthetic, or natural compounds investigation, we found anti-malarial drug chloroquine as an effective drug. So the road is not smooth. So for this plant, now about my research. For this plant, Drabanamorosa, our goal was to isolate compounds having cytotoxicity for cell line. Cytotoxicity for cell line. I used a 3 cell line. And we discovered 6 compounds indicated here, 1, 2, 3, 4, and another 2 is in another pages. So we discovered 6 compounds having excellent activity against all the cell lines. And interestingly, its activity is in nanoscale. See, number 29 compound, number 29, its activity is around 10 to 15 nanogram per ml. Whereas standard anti-cancer drug and standard cytotoxic drug, cisplatin, its cytotoxicity is 10 microgram per ml. This one is 10 nanogram per ml, and standard cytotoxic drug cisplatin is 10 microgram per ml. That means 100 times, 1000 times more active. So this compound is 100 to 1000 times more active, this compound, than standard cytotoxic drug. Getting this information, professor was very happy, and he thought a big dinner party, cocktail party. Why? Dreaming he is going to be a billionaire. Why? Because, you know, if we discover a cytotoxic drug, and if we sell these drugs to pharmaceutical company, he will earn billion dollar for next 10 to 20 years. And this is a general thing. So professor was very happy, and whole night party. But that time we did not get the appropriate structure of the compound. Just we isolate, and we tested against cytotoxicity, and we found its activity in nanoscale. So professor was happy and threw a party. So after a few days, we were able to determine the structure using NMR techniques. And after getting the structure, I was hopeless. Professor was also hopeless. Why? Because I found in internet in 2006, I found each year around 100 horses are dying after ingesting this type of plant. Actually, the plant name is foxglove in India. In India, around 100 horses are dying after ingesting this plant because the compound were digitalis group of drug, cardiac arrest. Due to cardiac arrest, 100 horses are dying each year. That time, 2005 data. And in northern part of India, I got that data. So due to the presence of the cardiac glycoside, digitoxygenin, you know, the leaves, flowers, and seed of this plant are poisons. So the leaves, the flowers, the seeds of this plant, all are poisons, and all are poisonous to humans and animals, and can be fatal if ingested. So what is our goal? Our goal is to isolate a compound which is safe for human being or mankind, not to get excellent activity, but that should be beneficial for human being. So my professor, after knowing this story, he was hopeless, and ultimately my pressure was increased. It's called fate. But remember, fortune favors the brave. So I started again. These are the compounds, digitalis group. That was I just made a paper, interesting papers. That's it. The NMR data of these compounds, antioxidant, and you know cyanic acid derivatives, cinnamic acid derivatives are very good antioxidants in nature. So we got some good antioxidant properties and antioxidant activities. Sometimes some antioxidants are similar to ascorbic acid. Number compounds, say ascorbic acid antioxidant activities millimole, 11 millimole, whereas my compounds 17, number 17 compounds activity, which is a cinnamic acid derivative around 15. That is similar to ascorbic acid. So we got good antioxidant active compounds from this plant. Cytotoxicity acid. See the cytotoxic cardenolides are 1000 times more active than standard cisplatin, but no output is zero because it is not good for human beings, and the compound is known or unknown. I got one new compound among these 15 or 16 compounds I found. One was new one. Okay. So discussion from Java, we found two novel antioxidant synaptic acid derivatives. One new cytotoxic campherol glycoside and on cardinolides. And also cardinolides are first reported from this genus. Though traditionally this plant has been long used as expectorant, stomachy, diuretic, anti-carcinogenic and anti-inflammatory in Korea as well as in Chinese medicine, but since we know there are some harmful or poisonous substances, so I wrote here, although traditionally it is used as anti-carcinogenic agent, but more research should be carried out to find its safety dose. Two papers have been published in essay journals from this plant. Third and last plant was Trichosanthem Hplc. This one, Phanultari, it's a local name, Korean name. And this plant, from this plant, I also isolated around 12 compounds. Among 12 compounds, 2 or 3 were new one. This is the Hplc data. Isolated lignans. Isolated lignans. Actually isomer. First, third compound is a new one. I also isolated flavonoids. First flavonoid bordered by red color is a new one. Also isolated some fatty, lots of fatty acid was there. Other compounds, ketone derivatives. This one was also new one. And cytotoxic acid. We found a moderate cytotoxic activity of compound 40, new one. And since we know cisplatin's activity around 10 against A549, and my compound 40 activity 10 times more, around 1 microgram per ml. So this one was considered as an anti-cancer cytotoxic drug. After getting this cytotoxic result, we did perform actin polymerase destruction experiment, which influenced apoptosis. This experiment was performed in another lab, but I was involved there also. It is a type of immunofluorescence analysis. Using immunofluorescence analysis of epidermal keratinocytes, HACAT cells. Here we see actin filaments, red part, actin filaments, and nucleus, blue, microtubules. And this compound inhibit this type of polymerization. More they inhibit, more activity, anti-cancer and cytotoxic activity. See, actin polymerase at 12 microgram per ml. The compound 37 shows complete destruction of aggregated structure of destruction of actin polymerization as well as tubulin polymerization. So, we published this data in a very good journal, and that one is Bioorganic and Medicinal Chemistry journal. And now synthesis of this compound is going on in our PhD lab, in my PhD lab, Kongsberg National University lab, to find out more active compounds that inhibit tubulin and actin polymerization. Now discussion. So novel cytotoxic lignan, keto derivatives, and flavonoids were isolated from the seeds. Both lignan and flavonoid exhibited potent cytotoxic that rationale the traditional use of this plant against tumor. And lignan 47, right side first picture, shows activity against actin cytoskeleton similar to pallidin derivatives. Actin polymerase inhibitor, pallidin derivatives. That's it. So from this discussion, what did we learn? Just as I was discussing about the way to proceed to get phytomedicines. It is also called bioassay guided isolation from natural product or from herbs or from herbal medicine. So first thing is to proceed to get phytomedicines. Why phytomedicines? Because some are active against toxic cell lines, against oxidants, against fungi, as well as against microorganisms. So the process to get phytomedicine, we learn. This is the first thing we learn. Second thing, I showed some spectroscopic techniques, basically NMR. So we can learn spectroscopic technique to determine why, to determine the structure or to elucidate the structure. This is also a part of natural product chemistry. And third is the biological test to verify the activity of the isolated compounds. These first things we can learn from natural product chemistry. And fourth thing is publishing the paper for international recognition. Why we will publish the paper? For international recognition. And after finishing these things, what will be, after finishing this type of things, what will be our benefit? All of this, after finishing all of these things, there will be in vivo test, there will be clinical trial to get the final product useful for human being. I already told you, for this reason, Professor was very, very happy. 30 years ago, 40 years ago, when Professor was in USA, the time one scientist discovered Taxol. 1980 or 1965 to 70, that time, one scientist discovered Taxol. And then 1985 to 1990, it was marketed. Taxol was marketed. See, it's long history. 30 years ago, after discovering any natural product, to get the desired benefit, we have to wait. Why we have to wait? Just for our safety. Same thing is going on for COVID-19, because of safety, just for safety. It is active. Vaccine is active. Vaccine is effective, but we need a human trial. We need safety. And for safety, we have to wait to get the desired product for a human being, good for human beings. Anyway, that's it from me. Thanks. So is there any question or something? Oh, this is the lab members. These are the lab members. So, hello. Yes. Yeah, welcome. Hello. Is there anything? Okay. Yes. For what? For this plant? I mean, you mean what do we know? What why we are studying on natural product chemistry? Okay, basically things is, you know, we know in India there are lots of herbs, lots of Ayurvedic companies in India and in Bangladesh and also in modern developed countries that are herbal medicines. Herbal medicines are cheap, their effectiveness is really nice, and they have less toxicities. But the thing is, all of us knows the effectiveness, it takes time to get effectiveness of any herbal product. We have to wait. But when we use pharmaceutical, let's say for example you have a fever. When you have a fever, if you take a synthetic product, para-hydroxyacetaminophen, paracetamol, you will get instant decrease of temperature. But when you take herbs, it takes time to get. Actually, basically, natural product deals with all herbs that have folkloric reputation. So we, to get the desired and active principles, we are using this natural product chemistry research. But anyway, we can use also herbal things, herbal products.
Okay, sir. So there is a question in the chat box, sir.
Yes.
By Mr. Athanaswaran. Can you suggest a suitable and easiest method for isolation of flavonoids?
Okay, isolation of flavonoids. Okay, flavonoids. Actually, always remember what you are going to isolate in terms of polarity. So, if you think the compound are polar, then the method will be one way. And if you think the compound is less polar or non-polar, the method will be another way. Okay, so we have some common extracting solvents. Polar solvent, we know polar solvent, methanol or ethanol. In most of the cases, flavonoids contains what? Contains hydroxyl group, and sometimes it contains sugar, that is glycosidic flavonoids. To isolate compounds of flavonoid, especially flavonoidal compounds, if you partition, if you do direct extraction using chloroform, I think you will get almost all flavonoids that doesn't contain any sugar unit. When the flavonoid contains sugar unit, you could go with ethyl acetate. So in ethyl acetate, you can get flavonoids. If you do extraction with ethyl acetate, you could get flavonoids with sugar unit, as well as some flavonoids with the hydroxyl group. And I think ethyl acetate is a good method. For I did fractionation using ethyl acetate, and I found a lot of flavonoids like pectolilaginin, hispidulin, and other things. Thanks.
Thank you, sir. So there is another question. Is there any similarity between Indian or Bengali medicinal plants and Korean medicinal plants? So any commonly used medicinal plants are there?
Yeah, there
between
are
India, Bengal and Korea, sir.
Okay. Yeah, there are lots of medicinal plants common. Oh, there are lots of medicinal plants that are common in India and Bangladesh, not in Korea and India. Why? Because Korean environment is different. You know, India is a very big country, and Korean area is similar to West Bengal area, similar area, but it is covered by 70% mountain. And in winter season, its temperature is around -5. In my place, Gwangju, it was around -5, -6, -10. So lots of snow falling. That times, so actually the plants there is completely somehow different, and because the growing condition is different. So in India and and you can find this environment in Kashmir, also in Darjeeling area, mountain area. Darjeeling area's plants are similar to the Korean plants. And I found some a very few, not that much plants. Bangladeshi plants are in Korea, like mango tree, you know. But the problem is, when I took mango from Korea, I didn't find any taste. If you are habituated to take Indian mango, you will never want to get the taste of Korean mango. Why? Because that is some sour in taste, not sweet in taste, like what we desired. So based on actually environmental condition, plant grows. We know this. So their plant is somehow different than our plants. Thanks.
Thank you, sir. So there is another question by Mr. Aziz, sir. As per the taxonomy, which plant families contain flavonoids and phenolics, sir?
Okay, as per taxonomy, which plant contains, I'm sorry, plant I found Asteraceae, Verbenaceae, and other. You can see any basically flavonoids if it doesn't have any glycosidic linkage that are yellow in color, yellow in color, and it has a lot of protective effects. But which genus or which family belongs to, it is really hard to say. It is really hard to say. Why? Because thousands of species. I didn't categorize. I did not think this way. Because around 50 to 40 to 50% plant I found that have flavonoidal structures. Sorry, I didn't think that way. That which part contains, which taxonomical contains flavonoids. Thank you.
Thank you, sir.
Any other questions?
So since you have been actively involved in natural product research. So
Yes.
during this pandemic situation, any of the traditional practice has been used in Bengal, sir? Bangladesh? Because in India we have used a lot of traditional medicine for a prophylactic measure or prevention measure or somewhat enhancing immunity. We have used a lot of traditional medicines. What about the Bangladesh, sir?
Yeah, in our country, actually, in our country, we are not habituated to use herbal medicines. I don't know this is why, but we are using a small percentages, a very little percentages. Tribals. In our country, there are some tribals. There are some tribes. They are using completely. They are completely dependent on herbal medicines. They don't believe. They don't. I say they don't believe allopathic medicine. Rather, they use traditional medicine. We say herbal medicine, and in village, and in cities or towns, a few. Nowadays, though it is increasing day by day, increasing. For example, cough syrup, bhashok, cough syrup, and other a little, not much, a little products are used by peoples living in cities. Most of the peoples in cities, they depends on Ayurvedic medicines, but in villages, in tribes, a significant amount of people depends on herbal medicines, but it is growing. Herbal sector in Bangladesh is just a growing up sector, and day by day it is growing. And I hope within very short periods of times, it will be a significant number of people in Bangladesh will use herbal drugs. But in India, I think the India scenario is different. Many people depends on herbal products. Okay. Thanks.
Thank you, sir. There is another question from Dr. Regina. Sir, you have isolated many anti-cancer compounds from your experimental plants. Had all those checked for their effect in animal model for cancer already? If not, do you have any plan to do?
Thank you. Yeah, as I told, apoptosis in tubulin polymerase inhibition activity was done, and that one was, so far I know, on 2007 or 8, that compound was in clinical trial in Korea. We put the name Hanultarin, the name of the compound was Hanultarin, because it was discovered from Hanultari. Local plant name was Hanultari, Trichosanthum clearloci plant name. And that one was in clinical trial. And other, we isolated nice cytotoxic component from Draba nemorosa. But as I told, that one is just Digitalis group of drugs. So we didn't have any plan for that. And that's it. And for antifungal assays, so far I know, 2 or 3 compounds are taken by pharmaceutical industries. They are using locally. They are using that as an ointment on surface. They are using on surface against cycle and a dermal infection. So, and personally, I don't have any plan to proceed further. Why? Because I don't have scope. I cannot do that based on rules and regulation. If I want to do further work on that specific compound, I cannot do without the permission of my professor. And this is legally I cannot do. If I want to do legally, then I have to discuss with professor too. This is just different things. Okay.
Yes, sir. Thank you, sir. So there is another question. Even in your studies also, you have used large quantity of extract compared with the allopathic medicines, like the standard medicine, whatever you're using for testing comparison studies.
Hello, I couldn't hear. What? Yeah. Could you tell again?
Yes, sir, please, sir. Yeah. In your studies also, you took a large volume of extracts compared to the allopathic medicine, sir.
Yeah.
So, what is the need to prove that thing, sir? Because we are taking English medicines in small quantity, but our herbal extract is a larger quantity. So, then what is the need, what is the thing to prove in that, sir?
Okay. Basically, if you see the activity, let's say Draba, and you know, in Ayurvedic medicine, to get herbal drugs, we need to put at least 400 milligram a capsule. Let's say for a capsule for its effectiveness. From Draba, we found a very good cytotoxic compound. Let's say compound A. Its activity is nanoscale. If it was good for human beings, let's say, so it's nanogram scale. See, that one was 400 milligram. But if you have activity, if you have a specific compound which is active, your dose will be certainly reduced. And what will be dose depends on clinical study or trial. That is not my concern here. So important thing is, if you get directly active constituent from a herbal medicine, the desired effect, specific or desired effect, the probability of desired effect will be high. It will be always high. Why? Because this is a pure form of that medicine. Sometimes synergistic effect happens, but that is not common. And if you take herbal medicine, the toxic effect will be less. That's okay. We know the toxic effect will be less. But thing is, if we take pure part from that compound responsible for the activity, the desired effect, the probability of getting desired effect with time course of study will be less. That means within very short period of time, you will get the desired effect. But if you take direct herbs, you will get the desired effect, but it takes long period of time. Why? Because the concentration of the active drug in herb itself is very minor in amount. That's. Thanks. Anything else?
Yes, sir. That's all, sir. Thank you, sir. Thank you for your wonderful presentation, sir.
Welcome.
So you have shared your experience for our students. It's very nice to us.
Yeah, thanks.
Sir, so if you have a chance to visit India, kindly let us know, sir. We can
Yeah, thank
invite
you.
through our
Yeah,
institution. You can directly share your research experience. If there is any chance, we can able to collaborate some of the research work, because here we are having a lot of herbal sources. Like we are in the under the Western Ghats, which is one of the main source of herbal resources of India. So we can able to help you out, sir.
oh, that's great for me. Please keep in contact with me. We can. Nowadays I'm working on probiotic as well as this anti-diabetic work. So if any faculty member is interested, just we can share. We can do a collaboration work with each other. That would be great.
Definitely, sir.
Yeah.
Definitely, sir.
Thanks, sir.
Yeah, and one more request also, sir. If any of your friends and colleagues, those who want to contribute in our virtual learning series, kindly refer them, sir. So it
Yeah,
will be
you already.
students.
Okay. I already referred on molecular biologist. He's a professor, and I will introduce more. I guess. I hope.
Okay, sir. Definitely, sir. Definitely,
Thank you.
sir. Thank you, sir. Thank you very much, sir. Thank you for spending your valuable time with us.
Okay, bye.
Yeah, hope to meet you soon, sir. Thank you, sir. Bye, sir. Bye.